So if we're thinking like a pathogen, once it gets into the host, again, it needs to grow, and it needs to grow even in the presence of our immune system. So one of the biggest concerns for microorganisms in our body are the phagocytic cells. Phagocytic cells are white blood cells, such as monocytes, neutrophils, macrophages, that are able to engulf and destroy pathogens. So if you're thinking like a microorganism, you're obviously concerned about phagocytic cells. So some bacteria have what we call anti-phagocytic factors. Anti-phagocytic factors are things that they can do to avoid being destroyed by a phagocytic cell. So primarily there are three anti-phagocytic factors. So the first being a leukocidin. So some bacteria are able to produce this leukocidin toxin, which is able to damage white blood cells such as macrophages and neutrophils and essentially kill them, so that a bacterial cell would be able to avoid destruction by releasing the toxin to kill a phagocytic cell. Another thing is that some bacteria have protective capsules, which is what we're seeing here. The clear surface that's surrounding the bacteria, this is its capsule, and that's a protective feature that protects the bacteria against phagocytic cells. So if a bacteria has a capsule, such as Streptococcus pneumoniae, it's able to avoid phagocytosis. And that's why that particular bacteria is able to cause a very severe form of pneumonia. So when you get infected with that bacteria, the alveolar macrophages in the lungs, which are meant to destroy infectious pathogens in your respiratory system, are not able to clear the infection through phagocytosis. So the infection gets really bad, and oftentimes the only way to overcome it is through seeking antibiotic medication. Then the last thing is some bacteria can simply just survive inside of a phagocytic cell. So they do get engulfed, but they don't actually get destroyed by the phagocytic cell. So they are not susceptible to the digestive enzymes the phagocytic cell releases to destroy them. The best example of this is Mycobacterium tuberculosis that causes TB. This bacteria has a very waxy cell wall, so when it does get phagocytosed by a macrophage, and that macrophage releases digestive enzymes to destroy it, that waxy cell wall actually protects it, and Mycobacterium tuberculosis is able to survive in the macrophages for extended periods of time. So naturally, these are just various ways that a bacterial cell would be able to overcome the effect of a phagocytic cell on our body and continue to grow and divide. So assuming that it went in through a portal of entry, it was able to adhere to a cell or tissue, it was able to colonize and grow, even in the presence of our immune system, the next step is to release, or the next step is obviously to impact the host in some way. I mean, this can look a variety of different ways. Sometimes microorganisms have toxins that they release to affect the body. Sometimes they have enzymes. And while we're not going to focus on the third, because we'll talk about it later on in the semester, sometimes microorganisms invade your body and actually manipulate your body into attacking your body. This is common for something like Borrelia burgdorferi that causes Lyme disease. When you get infected with that bacteria, your body produces antibodies to fight the infection, but the antibodies that it produces to fight the bacteria actually cause inflammation in the joints and lead to severe arthritis. So the bacteria has kind of manipulated your immune system into attacking your own body in that case. And we'll focus on those specific diseases later on the semester. Right now, what we're going to do is focus on enzymes and toxins that bacteria can excrete that would affect our body in some way. So there are dozens and dozens of different bacterial enzymes that can be produced. I did pick the two most common. These are especially common for like Staphylococcus and Streptococcus bacteria. But the two enzymes are coagulase and kinase. So coagulase and kinase are bacterial enzymes that essentially allow them to play hide and go seek with our immune system. So to kind of look at it, coagulase is a enzyme that bacteria can release that is going to allow a blood clot to form around the bacteria. And they do this as a means to hide themselves from immune cells like macrophages. So here we have some skin tissue. There are some bacteria here in the skin tissue, and then we have a blood vessel right here. So these bacteria in the skin tissue would obviously be concerned that a phagocytic cell or other type of white blood cell would find them, so they want to hide. So what they're going to do is release the coagulase enzyme, and that is going to pull clotting factors out of the blood and form a blood clot around the bacteria. So now the bacterial cells are essentially hidden inside this blood clot. So if a white blood cell would arrive on site looking for bacteria, it wouldn't recognize that there's bacteria in here. It would just see the blood clot and it would just continue on, and the bacterial cells would be able to hide. And now eventually the bacterial cells want to break free. They don't want to stay in there forever. They want to break free to continue to spread throughout the cells, or spread throughout the tissue and potentially into the blood. So when they're ready to break free, when the immune cells are gone from the immediate area, what they're going to do is release a second enzyme called kinase. Kinase is an enzyme that can actually dissolve blood clots. So here, once they're ready to be released, they release the kinase enzyme that dissolves the blood clot, allowing the bacteria to continue to spread throughout the skin tissue, but then also get into the blood where they can spread through other organ tissues in the body. So again, it is essentially kind of playing hide and go seek with our immune system, but coagulase is the enzyme that forms the blood clot, whereas kinase is the enzyme that breaks down the blood clot. Many bacteria will also produce toxins. So there's two different classes of toxins that bacteria can produce, exotoxins and endotoxins. It's really important that you know which type of bacteria produce which type of toxin. So exotoxins are produced by gram-positive bacteria. Gram-positive bacteria can make a toxin and then secrete that toxin out of their cell membrane. So essentially they kind of just spray it out of their cell membrane. So exotoxins come from gram-positive bacteria. Endotoxins are different. Endotoxins are only found in gram-negative bacteria. Specifically, they're found in the outer cell membrane of gram-negative bacteria. In the outer cell membrane of gram-negative bacteria, there are lipopolysaccharides, which are just fatty sugars that the bacteria can release either when it's dying or dividing. And when it releases these lipopolysaccharides, they have toxic effects in our body. So we classify those as endotoxins. So endotoxins are released by gram-negative bacteria, whereas exotoxins are released by gram-positive bacteria. So we're definitely going to focus on exotoxins a little more. They're just more common. So there are different types of exotoxins. All of these are produced by gram-positive bacteria and are secreted outside of the cell. And they're really easy to differentiate between because their name kind of tells you how they function within the body. So the four that we're going to focus on are hemolysins, leukocidins, neurotoxins, and enterotoxins. So hemolysins are exotoxins that affect red blood cells. Red blood cells contain hemoglobin, so that's where we get the name hemolysins. So these are toxins that are able to damage red blood cells. There's two different types of hemolytic toxins, alpha hemolytic toxins and beta hemolytic toxins. Alpha hemolytic toxins are toxins that only partially break down the hemoglobin and therefore only partially destroy red blood cells. So there's a partial hemolytic effect where the red blood cells are only partly broken down. If you put a bacterium that has an alpha hemolytic toxin on a blood agar plate, we will see this kind of pale green growth. So that is how we would know a bacteria would have an alpha hemolytic toxin, because on a blood agar we would get this green growth. The other type of hemolytic toxin is a beta hemolytic toxin, which is much worse. Beta hemolytic toxins completely break down hemoglobin and therefore completely destroy red blood cells. If you put a bacterium that has a beta hemolytic toxin in blood agar, you're going to get these big clear areas in the agar where the red blood cells have been completely destroyed. A bacteria that has a beta hemolytic toxin is something like Streptococcus pyogenes, which causes strep throat. In fact, blood agars can actually be used to culture and diagnose a strep throat infection. Another toxin, we've already talked about this one, but it just comes up again, are leukocytins again. Leukocytins are toxins that will damage white blood cells, which often get called leukocytes. That's where the name leukocytin comes into play. But specifically, leukocytins will typically damage white blood cells, predominantly phagocytic cells, things like neutrophils and macrophages. So they really affect the immune system more. Neurotoxins affect the nervous system. And since the nervous system controls our muscular system, typically neurotoxins would cause things like muscle spasms or muscular paralysis. And something like the botulinum toxin that causes muscular paralysis, the tetanospasmin toxin that comes from the bacteria that causes tetanus, that's going to cause muscle spasms. And then there are enterotoxins. Enterotoxins act on the gastrointestinal system. So if you've ever had food poisoning caused by something like Staph aureus, you've experienced the enterotoxins. So it would cause vomiting, diarrhea, cramps, upset stomach, that type of stuff. So important that you understand what part of the body these different neurotoxins or different exotoxins target. So now that the pathogens are in our body, they have colonized and they are releasing things like enzymes and toxins to make us have a disease, we are going to start to experience signs and symptoms. We know we have a disease when those signs and symptoms start. So often the words sign and symptom get used interchangeably, but they do have specific definitions. They're different. So we should be able to differentiate between a sign and a symptom. So a sign is something objective. It's something that you can physically see or measure in some way. Like we could physically see a patient having a seizure. We could see if someone was sneezing. A symptom is subjective. A symptom is only felt internally by the patient, so they would have to kind of describe it to you. You wouldn't be able to see it or measure it. For example, pain. Pain is very much subjective. It's a symptom. It's something that the patient would have to prescribe. Or if they were fatigued or if they had a headache, we wouldn't be able to visibly see this. So to look as a review, something like a fever, that's more so a sign because we can obviously get their temperature and get like a numerical read on how high their fever is. Someone just says they have body aches and we can't see that, so that's going to be a symptom. Swelling would be considered a sign, so we can physically see it. Same with vomiting. We can physically see it, so it is a sign. Nausea, though, would be considered a symptom. We can't see that a person is nauseous, so that would be more so a symptom. Discomfort is a symptom. Diarrhea, obviously, a sign. Coughing is a sign, and so is having a rash. So do make sure you are able to differentiate between a sign, which is more objective, and a symptom, which is more subjective. So once a pathogen has kind of wreaked havoc inside of a host, it eventually wants to leave so that it can enter another host eventually. So inevitably pathogens will exit through a portal of exit, and their goal is hopefully to exit that host and then eventually gain access into a portal of entry into a new host. So the site where the pathogen leaves the body is simply called a portal of exit, and this is some type of blood or bodily fluid. Blood, bodily fluid, feces, all of those are considered portals of exit. So that's why you have to be incredibly careful with how you handle things like blood and feces and vomit and even mucus if we're talking respiratory infections, mucus droplets, because all of those are portals of exits that could potentially harbor pathogenic microorganisms that get into your portal of entry of the host and make you ill.